Hi everyone. Good morning, good afternoon, or good evening, independently where you are watching us from. Welcome to our Aiforia webinar from today. Today we'll be exploring breast cancer diagnostics and how AI can enhance clinical decision-making through more consistent and efficient image analysis. My name is Ruth Pedrosa and I'm a Senior Field Application Scientist at Aiforia Technologies. I'm very pleased to be joined by Dr. Richard Doughty, who will be contributing to today's discussion. Before we narrow our focus to breast cancer, I would like to start with a brief introduction of Aiforia and our solutions. Aiforia is a Finnish medical software company with deep roots in pathology and image analysis. We provide pathologists and scientists with powerful cloud-based deep learning AI technology. We work with hospitals, pharma companies, and research institutes in over 50 countries, and our tools are used across many disease areas, from oncology to toxicology. Our headquarters are in Helsinki, Finland, with subsidiaries in the United States and France. At the heart of everything we do is our mission to transform pathology image analysis with AI, enabling better care for every patient. Aiforia's portfolio spans the full journey from research to clinical practice. Aiforia Clinical Suites deliver complete, case-centric workflows designed to enhance efficiency, consistency, and diagnostic accuracy in pathology labs. We currently offer the broadest portfolio of CE-IVD-marked AI models for diagnostic support. Our research solutions, on your right side, provide fully automated study-centric workflows and image analysis applications suitable for all research domains, with full compliance to GLP standards. And finally, Aiforia Create, our flexible AI development platform, empowers users to design, train, and validate their own AI models. If your goal is to translate your new developed model into clinical use, Aiforia can help you with that as well. This slide provides a high-level overview of the Aiforia Clinical Suites, our CE-IVD tools that support pathologists across multiple diagnostic workflows. On the top left of your screen, you can find our Lung Cancer Suite, which includes automated PD-L1 scoring. Just below that is the Breast Cancer Suite, offering H&E-based grading and complete IHC or immunohistochemistry panel, ER, PR, Ki-67, and HER2. Our Breast Cancer Suite will be the main focus of today's webinar. Continuing down, the Colon Suite features QuantCRC, our prognostic AI model for estimating the risk of colorectal cancer recurrence. At the bottom left, the Prostate Cancer Suite supports Gleason grading groups and the detection of key adverse patterns, including cribiform architecture and PNI. On the right side of your screen, we highlight two additional models. The lymph node metastasis model detects metastases originating from several primary tumor types, helping reduce variability in nodal assessment. And beneath it, we have our Gastric Suite, enables detection of gastric carcinoma and H. pylori on routine histology. Together, these suites demonstrate that Aiforia augments the diagnostic process, offering reproducible quantification, reducing subjectivity, and supporting faster case turnaround. And with that in context, we'll now shift our focus to the CE-IVD Breast Cancer Suite and how it can be integrated to optimize your existing workflow. Now I would like to hand the floor over to Dr. Richard Doughty. Richard is Aiforia's medical director, responsible for ensuring the medical accuracy and clinical relevance of our AI-powered pathology solutions. He brings extensive experience as a medical doctor with a strong background in pathology and diagnostics. Besides Aiforia, he works at Oslo University Hospital as a consulting pathologist. In addition, he is a qualified veterinarian with pathology expertise in pharmaceutical research and development. Without further ado, Richard, over to you.
Hi, everybody. It's a great pleasure to be able to talk about workflow in AI diagnostics and eventually highlighting our breast cancer suite and our models that are available to um, integrate into your diagnostic workflow. Just a little bit about the agenda that we will have. We will talk a little bit about why workflow matters. In pathology, we will take a broader perspective of what has been learned from other industries, such as the automotive industry, radiology, and clinical practice. We'll then talk about what the current breast biopsy workflow is and where its bottlenecks are, and then how AI and specifically how Aiforia's tools can help with those workflow challenges. We will also quickly touch on the lymph node metastasis detection system, which can be part of the diagnostic workflow for breast. And then we will talk about how the models can be used to triage, reflex test, and make your workflow more efficient. In the end, we will talk about the end game or the long-term vision of how AI may assist you in your daily practice. Why does workflow matter for AI? And I'm going to take a very general view and then gradually focus down to our products, Aiforia products, because I think really for us pathologists or lab directors, it's really important to understand why it's so important. And workflow really is one of the most crucial elements. We've all heard the buzz about AI's potential to transform our field, yet in reality, many tools haven't made it into routine use because they don't fit smoothly into how we work day to day. In fact, despite sort of tremendous technical progress, real world AI adoption in medicine has actually been quite limited, often not because the algorithms don't work, but because of the workflow issues. So this is really crucial. A really top-notch AI algorithm that forces clinicians to change or add tedious steps can end up unused. A high-performing algorithm, on the other hand, can really fail practice if they disrupt the existing routines. People are used to what they're doing. And if they suddenly have something that becomes difficult or acts as a pressure point, they will bypass it or ignore it. On the other hand, when an AI tool is seamlessly woven into your daily workflow, it truly can deliver efficiency and safety gains in your clinical practice. In short, how we integrate the AI is just as important as to what it does. So in this first segment, we'll explore a little bit about what we've learned about the importance of workflow in AI integrations in other industries. We'll draw them from things like healthcare in general, AI, or the aviation industry, the automotive industry, and then we'll break down how this workflow can really make or break the success of AI in clinical practice. And if you look here, you're wondering probably what relevance does an airplane have to our to pathology? But really, in aviation, automation has been a game changer. Autopilot and other AI systems now handle routine flying, dramatically improving safety and reducing pilot workload. But what we saw was a stark warning when the Boeing 737 MAX, which this is a picture of, had some accidents. And this was when an automated system was adding without pilots fully understanding or training in the system that was being added in. The result was two really tragic crashes. The system's design flaws and the lack of pilot awareness led to disaster. The lesson here is that even in aviation, if automation isn't transparent and thoughtfully integrated into the pilot's workflow, then it can backfire catastrophically. And that's the same lesson we can have in pathology. If we don't think about how it's going to be integrated, we're going to have problems. Other example is, for example, manufacturing. If you think about General Motors, the car manufacturer in the 1980s, they tried to pour money into what they essentially called a lights out factories filled with robots aimed at eliminating the human element. But they essentially bolted robots into an assembly process that was made for human workers. And they didn't really rethink the workflow or engage the workers in how they were going to implement it. The outcome was sort of infamous. The robots started to paint each other, they started to misassemble cars, and the productivity dropped. And the massive automation investment that they made never realized any cost savings. Meanwhile, if you take another company, automotive company like Toyota, that took a different path, they integrated the automation fully with a focus on workflow efficiency and human oversight. And this empowered Toyota, for example, to improve processes and use automation to a system and not replace the human. The result was a world-class quality and efficiency. So the manufacturing world learned that technology alone isn't a silver bullet. It must go hand in hand with the process redesign and the culture it supports. Also, finance and software have really experienced the same. If we go sort of home in a little bit closer to home in the healthcare arena, probably one of the most uh, interesting stories about technology integration, with also some cautionary tales, has been the rise of electronic health records. No one would dispute that EHRs have brought benefits like better data accessibility and legible records, but they also introduced a huge documentation burden. Doctors and nurses now spend a staggering amount of time just clicking and typing. By some estimates, one third to one half their workload in a day is taken up by EHR activities. That's time not spent with patients and arguably irrelevant time for a clinician. This is a workflow problem. Early EHR systems weren't often designed for daily clinical routines in mind. So they interrupted the clinician's workflow and added friction. The result? Frustration, slower workflows, and even burnout amongst the clinicians. When a system forces a clinician to spend evenings filling out forms or navigating clunky interfaces, you know the workflow's integration has failed. For example, a typical with EHRs is that a clinician may wait till the end of their shift to then work after their shift to fill out the paperwork. So workflow matters. It doesn't matter how good your AI is, it has to be fitted and integrated into your workflow. Because what happens when a tool doesn't fit well? Clinicians find workarounds. We've all seen this, writing notes on paper or maybe scanning them later in or having a separate sort of spreadsheet for your patients list because the EHR can't work. The usability issues have indeed led to rampant workarounds, duplicate documentation, copy pasting, using, as I said, personal spreadsheets, which in turn create new opportunities for error and inefficiency. In other words, if technology makes the workflow harder, people try and circumvent it, undermining the very goals of the system. It's a big lesson. Just implementing a new technology isn't enough. It has to mesh with the clinical workflow, or it might do more harm than good. A really good example of that, and it's not really related, but just to highlight it, would be the World Health Organization surgical safety list. This isn't high tech, it's just paper, but it really fits that if you find something that can fit into workflow, it can be really magic on how it transforms. What they found by standardizing these safety checklists on a form, hospitals worldwide saw a post-operative complication rate drop by a third and surgical deaths drop by about half. And that was just due to the implementation of a 19-item checklist. And what they found was there was a downtime between when the patient was coming to surgery and having the surgery that they could fit this checklist in. And it integrated into the surgical workflow perfectly. It was quick, it happened at a natural pause point, and it addressed a real problem. The success story tells us that sometimes the solution isn't as fancy as AI, but if you get a smarter workflow, it helps you. Process matters. This is just coming nearer to home to radiology and image-based specialists, which often we take a lot of information and a lot of what they've learned from radiology and AI over to us. Now we're starting to try and AI our processes, and radiology has really been at the forefront of AI implementation. It's been something of a sort of testing ground for AI in healthcare with dozens of algorithms for detecting nodules, fractures, hemorrhages. In fact, I think the FDA has over 1,000 approved apps for radiology compared to just maybe a handful for us in pathology. So there's a lot to learn from them, and that's why really I'm focusing on these before I focus down to our Aiforia breast models. This early experience has been enlightening for us. It's shown that radiology achieved some integration, but not without overcoming major workflow hurdles. What were those hurdles? The first was fragmentation. In recent years, radiologists have been courted by a myriad of AI tools, one for lung node detection, one for flagging strokes, another for measuring bone age, etc. Each tool often came with a different vendor. Early on, departments found themselves having to sign multiple contracts, install multiple separate systems for each narrow use AI model. Imagine the headache. You might have a fracture AI from vendor A, a pneumothorax AI from vendor B, each with its own software interface. Integrating these into the radiology IT environment was costly and complex. There were no universal standards, and this made it very difficult to plug AI into the radiology workflow and scale it up to a level of usage that was clinically useful. Radiology departments realized really quickly that without a more unified approach, these fragmented tools would remain niche pilot projects and just a curiosity that was often not used by the radiologist. Secondly, there was no clear workflow playbook for AI. Even every hospital was doing their own thing. One might use AI for pre-reading mammography, the other might just use it for research. The use of AI was highly variable and ad hoc, essentially. Because of this, it was hard to gather evidence of its value to support its use, which was almost like a negative spiral. If everyone is using the algorithm differently or sporadically, how do you prove that AI improves radiology practice? Some sites saw efficiency gains, others didn't, often depending on how well the tool was integrated into the workflow. This variability made it challenging to justify the broader adoption or to identify what best practices in radiology were. Radiology lacked the standardized guidelines on how to implement the AI in reading workflows. The next was really people factors, and I think this is one of the most important. All of us, when we use AI, are interacting with an interface, and radiologists found themselves, they had a learning curve, and this is the learning curve we're having at the moment. Many practicing radiologists never formally trained in AI or machine learning. Surveys and reviews found that radiologists had a really wide, broad range of expectations about AI. Some feared they weren't going to have a job. Others thought it was a magic bullet, and many lacked a basic understanding of how these algorithms work. The literacy sort of gap meant that some radiologists didn't trust the AI, worrying that it was a mysterious black box, while others were over trusting it without really critically analyzing what the result was. Building a realistic, informed mindset in the user base emerged as a necessary step to the adoption of it. So in your departments, you may have pathologists who are absolutely on board with this AI. They think this is going to be the future. There are others that may be more in worrying about what does it mean to me? How will I use it? How do I use it? Is it going to me just approving it? Is it going to be me working with it? So these are really important factors that need to be addressed in every department when they decide to use an AI. Relatedly, early AI systems in radiology often suffered this sort of black box workflow issue. Typically, an algorithm would analyze an image and present an output, saying, for example, highlighting a lung nodule. But the radiologist had limited ability to interact with that output, and that was quite worrying for the radiologist. For instance, if an AI circled a region, the radiologist couldn't easily adjust it or give feedback through the system. They either had to accept it or just ignore it. And this often, if they thought there was a performance issue, just led to them ignoring it and not using it. There was no immediate loop for the radiologist to correct the AI or for the AI to learn from the radiologist's input. This lack of flexibility and transparency, essentially the AI acting as a one-way black box, undermined trust. Radiologists are trained to be meticulous and in control of the interpretation process, the same as us pathologists. And if an AI tool doesn't allow them to verify or change or edit the findings, it can feel very unsafe and at least unwieldy for the radiologist and the pathologist. Many radiologists responded by being very cautious about the output, double checking everything, sometimes turning the tool off, which of course reduces any efficiency gains. So to get efficiency gains and really understand how to effectively use AI, this has to be understood. So in summary, with AI, an early AI journey hit obstacles familiar with other domains that were seen in other industries. Integration difficulties, lack of standards, inconsistent use, user skill gaps, and trust issues. So how did they eventually sort of come over this? I mean, because they haven't just encountered problems. Radiology, as with pathology, is a solution-based speciality. And so how did they find solutions that we can learn from when we're designing AI for you pathologists, so you will have a seamless workflow? First, radiologists insisted on a seamless user interface for AI. Early on, some of the AI tools would require launching a separate application or web page to view the results, forcing the radiologist to switch screens, log onto other systems. This, for radiologists, was very suboptimal. It interrupted how they flowed, the reading of images flowed. Leading radiology department learned to embed the output directly into the PACS systems, which is their image handling systems. For example, one hospital, the leadership was adamant that a quick web link to an AI result was not good enough. So what they did was they worked with the vendors to integrate the AI finding, so that, say, AI tumor detection is highlighted on the same PACS image viewer when the radiologist is using it, avoiding extra clicks. There's evidence to suggest that if you have to click more times, a clinician has to click more times, they try and go around the system. By making the AI essentially invisible as a separate software, just another layer of information within a familiar tool, it reduced workflow interruption. Secondly, radiologists' sites began adopting a vendor-neutral platform, and instead of installing or maintaining a dozen separate different types of app, they set up an integration hub, often called a sort of AI orchestration platform. This platform takes images from scanners or PACS, automatically routes them to an appropriate AI algorithm, and then returns the results to the radiologist screen. It also centralizes things like logging, user management, and legal agreements. The third factor is the recognized need for new workflow roles and expertise. One innovative approach was creating imaging process groups, which made up of tech-savvy radiographers and technical physicians in the department, and their job was to deploy and integrate the advanced technologies and operate them as part of the workflow. Another strategy, of course, is keeping the human in the loop of the AI and making the AI outputs modifiable. This is something at Aiforia that we've been really working quite heavily with, that the Aiforia clinical viewer doesn't just give you an answer or a diagnosis or suggestion, it also enables you to actually interact, adapt, select areas, and get new results. So the modifiable output is important. Radiology tools increasingly allow radiologists to interact with the AI suggestions. For example, if an AI outlines a lesion in an image, the radiologist can adjust it or reject it if it's not accurate with a single click within the workflow. This way, the radiologist remains the final clinical decision maker, which is really important. And the AI serves as an assistant that can be corrected. All these strategies from radiology boil down to a common goal: making AI helpful and a natural part of the workflow, rather than distracting add-ons that take time and are irritating to click into. By doing hard work of integrating, standardizing, giving role definition and cultural change, radiology is gradually unlocking AI's promised benefits, but it's been through a long process of understanding that it's not just a model that matters. And as we see, these benefits are indeed emerging and even challenging, and potentially still remain. But it's important for us in pathology to understand them, what has been learned from these industries, and not make the same mistakes again. So with this improvement in workflow integration, what are the results that really radiology have seen? And they're really encouraging. Basically, what it tells us is when AI integration is done correctly, you get success stories, faster diagnosis, workload reductions, but it requires validation and refinement. There is a progress to celebrate, and at the same time, a recognition that we're still in the early days with many of these tools and still learning. The hype hasn't fully translated into routine clinical practice everywhere yet, and that's okay. It's a new technology. We need to learn together, but it underscores why focusing on workflow and real-world testing is so important. Radiology's hard-earned lessons can guide other fields like our own, pathology, to avoid pitfalls and accelerate the path from AI promise to the clinical reality. So what are the main lessons that have been learned? The main lessons really are seamless integration, not adding on, not individual apps, adapting process, not just bolting things on, reworking tasks and roles to maximize the AI value, keeping the human in the loop. Automation should augment the professional, not replace judgment. Maintaining oversight and control is really important for the medical professional, and solve real world problems, real problems that the pathologist has or the radiologist has, not just a sort of gimmick essentially. And also really what the fun part of this AI adventure in radiology and pathology is that it really focuses on that we need to invest more in people and the culture in the work environment, training users, building literacy, encouraging physician tech collaboration, and ensuring leadership that these the radiologists and the pathologists are leading the way and showing they're not just being given the apps and said use them, but they're also interacting with the technologists on how can we make this better. So now I'm coming probably to the bit that you have been waiting for, which is really how can AI help the pathologist go through a breast biopsy? And I just think it's really important first to look at what the standard workflow is today. And I understand that labs in different areas will have different numbers as to how long things take. But this is just a general view, for example, from a series of labs that we've had interaction with and the timelines they have. So the pathologist first examines the H&E stain slide that has come from a biopsy that's been taken due to the triple diagnostic or basically a patient has found a lump or a lump has been detected by the physician and they've sent them for an ultrasound, a mammography, and subsequently an ultrasound guided biopsy. When the pathologist gets that biopsy, the initial review is to confirm the presence of tumor or not, the invasive carcinoma. If it isn't evident on the first section, the lab needs to the pathologist needs to order some recuts on deeper levels, which introduces delays as additional slides are prepared and examined. But once the invasive cancer is identified on the H&E, the pathologist then orders a panel of immunohistochemical tests. And these are typically a standard battery test, estrogen receptor, progesterone receptor, HER2, and also KI, often KI67. These immunohistochemical stains usually run sequentially after the H&E review by the pathologist. So the pathologist looks, he or she sees tumor, and then they order the immunohistochemistry, meaning that the case now waits in the lab for a day or more while the slides are stained and processed. This stepwise approach inherently causes idle time. The turnaround time grows with each successive test. A real pinch point in breast biopsy pathology diagnostics is the HER2 test. By immunohistochemistry, HER2 often comes out as a 0, a 1 plus, a 2 plus, or a 3 plus. If the result is clearly negative, for example, 0 or 1 plus, or clearly positive, 3 plus, then the pathologist can finalize that result. But if it's equivocal 2 plus, which happens not infrequently, the protocol mandates that there needs to be in situ hybridization HER2 to check for HER2 gene amplification. This reflex adds significant delays. Often it can be up to a week or two, depending on the lab. In many settings, such as sending out, if you have to send it out to a secondary provider or to a molecular lab, a commercial molecular lab, it can take up to 7 to 14 days, prolonging the diagnosis and treatment planning. Meanwhile, the patient and oncology teams are waiting for this critical piece of information, which will inform them regarding chemotherapies. All of these serial steps, the initial H&E, deeper cuts if needed, the ordering immunohistochemistry, and then ordering possibly in situ hybridization, extend the overall turnaround time. It's not uncommon for a complex case with a HER2 reflex-ish to stretch across multiple weeks before the final report is ready. Another challenge in the traditional workflow, the manual workflow, which is using digital but non-AI or light microscope, is that many assessments are manual and subjective. The pathologist must determine the Nottingham grade of the tumor by eyeballing features like tubal formation, nuclear polymorphism, and mitotic count. Different pathologists may assign slightly different grades to the same tumor based on visual estimation, introducing inter-observer variability. Also, we know if you give the same pathologist the same slide after a number of weeks, you will also get intra-observer variability, where the pathologists may not be consistent within themselves. Similarly, regarding reading the immunohistochemistry, things like KI67, also the progesterone and the estrogen and the HER2, these are subjective often, especially near the borders of grading. These subjective interpretations mean that consistency can suffer. One pathologist low positive might be another's negative. In summary, the standard workflow is linear and time consuming, with built-in delays at each decision point and a potential variability in the results due to manual interpretation. These are exactly the pain points we want to address and want to make more efficient and more accurate with AI. As we discussed in the earlier segment, our current pathology workflow has several key problem areas that the AI implementation can target. First, the workflow and toolset are often fragmented. A pathologist might use one system for viewing a clinical viewer and another system, a list system for reports. This means that these tools often don't talk to each other, and this fragmentation can also force pathologists to manually bridge gaps, for example, by transcribing results or switching context between platforms. Secondly, the traditional workflow is highly sequential. Each step basically happens one after the other. So that means that pathologists are often finding themselves waiting. You can't interpret an immunohistochemistry stain that hasn't been done yet, and you won't order it until you see something on the H&E. This means there are built-in periods of inactivity on a case, stretching the timeline. We identified these delays as a major friction point. Conventional immunohistochemistry and also in situ hybridization testing accounts for a large proportion of the turnaround time in breast cancer diagnosis. Third, many steps are labor-intensive manual work. Think about counting mitotic figures and looking for them across the slide, or manually measuring the percentage of cells that are Ki67 positive. These are repetitive tasks that consume a lot of the pathologist's time and concentration. Classic bottlenecks where humans spend effort that an automated system might be able to handle faster. Manual work is not only slow, but it can also be error-prone and inconsistent. One person's count might differ from another's, especially at the end of a long day when you're tired, for example. That ties into the fourth issue, which is of subjective variability. Different pathologists might grade the same tumor differently or have a slightly differences in the way they interpret the staining intensity. I know myself, for example, the intensity of what I consider positive is often different to others. Some pathologists need an intense brown staining to be counted positive. Others are just looking for a very specific but light brown staining, for example. This lack of standardization can impact patient care. If one lab calls a case HER2 1+ and another calling it 2+, it may radically change the patient's treatment. Moreover, variability complicates lab operations. If results are inconsistent, sometimes tests get repeated and additional opinions are sought, further delaying things. Finally, the net effect of all these issues is extending the turnaround time and a heavier workload on the pathologists. They are working in an environment where globally the number of pathologists is not growing fast enough to meet the rising cases. In fact, in some regions like the US, the pathologist workload is shrinking even as workflow is increasing. Inefficient workflows exacerbate this strain. This recap of challenges sets the stage for why Aiforia is introducing AI that we hope tackles the workflow problems and integrates in a way that makes you as pathologists more efficient. I just wanted to quickly cover the idea of there are many ways when you're developing an AI model, the way you address them. And you can have them as first reader, and that means that the AI system doesn't analyze the case, but it can also, for example, pre-fill the report with what it suggests, and then the pathologist looks at it and essentially adapts it and approves it. The second is you can use it as a second reader, which is where you've looked at the case, you've put out, I just want to use the AI to check I haven't missed anything. You can also, of course, and these aren't independent with each other, use these first read and second read systems as triage, a way of sort of identify where are the cancer cases in this stack of 50 cases. You know, I want to prioritize the 5 cases that are cancer, but also screening where maybe in the future you don't have to look at the negative cases. I think it's important to understand that as a first reader system, which is what the Aiforia system uses, it saves work time, but it can also, if you have it it can reduce cognitive burden. Instead of starting, for example, from a blank slide and building up the findings from scratch, building up your diagnosis and let's say, for example, your case report, The pathologist sees pre-analyzed results and visually highlighted regions, helping them focus their attention more efficiently. In practice, this can mean faster sign-out, less fatigue from a routine case, and a stronger sense of control over the complex. In contrast, a second reader system is usually deployed after a full human review. It's valuable as a safety net, catching oversights and prompting re-reviews, but it doesn't help with triage so much in early test ordering or, for example, report drafting. And the human still does the full pass interpretation, so the cognitive load remains unchanged. This is why in Aiforia we've decided to have essentially a first reader system that can be used in triage as well, because we believe that the first reader systems provide more end-to-end workflow support, from case prioritization to AI-supported report generation, while the second reader system supports your error detection. The first reader AI improves efficiency, consistency, and also reduces mental workload. Key advantages in high-volume diagnostic settings, such as pathology labs. And these are the products we have offered or have developed for the breast workflow. And we believe we have the best integrated and most full offering for breast diagnostics. And this goes because we offer products from the beginning, where you're going to be identifying is cancer there, how to grade it. Also, all the immunohistochemistry biomarkers, the HER2, breast cancer, the estrogen receptor, and also the progesterone receptor. We also have Ki67. And how can these be used? Let's turn to the solution, starting with the AI model for breast cancer grading. This is a AI-assisted tool focused on on the HE slide. And it will come in here at the beginning. You get the HE slide on your digital system, and you open it up. The Aiforia breast grading has already been run and is ready for you to look at. And here you look, you come in the morning and you see your case list. You can see here that you have some lung cases, some breast cases, maybe a colon case here with the QuantCRC. But you can see here what it has done is it listed which cases invasive carcinoma has been identified. So this enables you to prioritize, say, okay, I want to deal with my breast cases. I want to look at only breast cases today. That's what I've been assigned. And I want to initially focus on the invasive, only the cases where invasive breast cancer has been identified. So I can prioritize those and get them signed out and get that treatment as quickly as possible. And this work list allows you to prioritize these, identify and select which are your priorities. And then once you get your slide, you click on it. And the very first step in our workflow, what does it do? In simple terms, the breast grading model, identifies the digitalized H&E whole slide image of the breast cancer and automatically determines the Nottingham grade of the tumor. It analyzes the same features as the pathologist would, how many mitoses are present, how abnormal the nuclei look, the nuclear polymorphism, but how much the tumor forms the glandular or the tubular structures. By evaluating these, it generates components of the Nottingham score and thus gives an overall grade. What immediately benefits is speed. In our clinical validation, using the AI grading model significantly reduced time needed to assess a slide. In fact, grading could become very rapid when the pathologist became very familiar and confident with the model. So roughly about 50% faster compared to manual reviews. The algorithm rapidly scans the entire slide for mitotic figures, which can often be hard to find, and other features much faster than a human looking through multiple high-power fields. This means that the pathologist can get a preliminary grading assessment in a fraction of the usual time, focusing their expertise review more efficiently. Importantly, this speed doesn't come at the cost of accuracy. The model's results show a high concordance with expert pathologist assessment. In other words, what the AI calls a grade 2, an experienced breast pathologist would also call a grade 2 in the vast majority of cases. By providing an objective score for each component in the grade, the tool helps standardize the grading process. This greatly reduces inter-observer variability, so between the pathologists in a department, their grading will become more consistent between themselves. Two pathologists using the tool are much more likely to arrive at the same grade than if they weren't using the model. The outcome is more reproducible diagnostics for a patient, regardless of who or where they're being evaluated. So if you have one hospital system with multiple pathology labs, but you're using the Aiforia breast grading, you will get a much tighter, much more reproducible diagnosis between those labs and between the pathologists within those labs. Another key advantage is how the model integrates into pathologist workflow as a supportive tool. The AI just doesn't spit out a grade in a black box fashion. It also provides a visual evidence at the cellular level of what it's finding. This gives explainability to the pathologist and understanding what the AI model has identified. For example, it can highlight exact cells. Is it counted as mitosis or mark regions with poor tubular formation, for example? And the pathologist can look at it and see if they agree with it. If they don't, they can change it. This transparency means that the pathologist can verify and trust the Aiforia breast grading model output. Essentially, the tool shows why it thinks a tumor is grade 3, allowing the pathologist to agree, override, or edit as needed. The pathologist have responded positively to this glass box rather than a black box features. It helps them understand and concur and also learn with the machine, with the model, how the model is thinking and what is the weaknesses, the strength of the model, and how that can be applied to that pathologist practice. In practice, the breast grading Aiforia model serves as a first reading assistant. The moment the H&E is scanned, the AI is beginning to analyze it even before the pathologist sits down to review the slide. By the time the pathologist opens his case, the AI identified the area of invasive carcinoma and generates a preliminary grading, which is supported with the annotations. The pathologist then reviews these results, looks at the highlighted areas, and uses their expertise to finalize the grade. This synergy of AI speed and consistency with human eye oversight improves accuracy and confidence. It's supporting the diagnostic workflow by making a very first slide from the patient to H&E more informative and quicker to interpret. Ultimately, this means more consistent grading for the patients and potential time savings in the lab, as cases can move quickly to the next step. This is just an outlook of the interface that the pathologist will see. As you can see, it gives you an image. This is the biopsy. The boxes are the mitosis, where mitosis have been identified, the 10 high power fields. And then you can click on and you can see, for example, what proportion of the pleomorphic score, nuclei score is, which is the highest. In this case, it would be score number 2. Then it will give underneath what the grade is, but it will also break down mitotic rate, nuclear pleomorphism, and the tubular differentiation, so you can see how it's come to that score. It also in this system will give ductal carcinoma in situ. So coming on to the next part of the diagnostic process, in addition to the H&E grading, Aiforia also offers immunohistochemistry models for the estrogen receptor, progesterone receptor, HER2, and Ki67. And these are really the sort of workhorse biomarkers of breast cancer diagnostic. These models tackle the next step of the workflow, the evaluation of these specialized stains. For estrogen receptor and progesterone receptor, the AI examines the IHC slide and identifies the tumor cell nuclei that are positive and negative. It then quantifies the percentage of tumor cells that are ER positive or PR positive. Essentially, it's automating the manual task where the pathologist might eyeball, about 80% of the cells are ER positive. Instead, the AI will count and give an 82% positive, for example, with great precision. So it takes this precision onto another level. It's also consistent. So if you look at the slide today, you will get an 82% and do it, let's say in a week, you will get the same. Moving on to HER2, it's a more complex marker because the score is about the intensity of the membrane staining. In tumor cells, the percentage of the cells showing the staining The Aiforia model is designed to mimic the CAP or ASCO scoring to effectively a 0, +1, +2, or +3, based on the membrane positivity. The AI analyzes each step, each membrane staining, completeness, intensity, and then computes the proportion of the cells in each category. By doing this, it provides a preliminary HER2 score from the slide. One big advantage of this is the consistency and standardization. I just wanted to show you really what the product does and how it looks. So I took a screenshots from from one of the interfaces, and here you can see, for example, you can see that this is, I think this is a PR stain, and you can see the brown staining, you can see the blue negative staining, and then when you run it through the model, you can see that the blue, the negatives are highlighted as blue circles, and the positives are highlighted as red. So the pathologist can look through and think, do I agree with this? Is it staining what I would think is a negative or is a positive? Also, just as an add-on, I just wanted to mention that we also have an Aiforia model that can identify lymph node metastasis in melanoma, colorectal, and breast. So this may come on later on when they might do a sentinel node resection, or for example, the axilla with breast cancer, and they might want to identify metastasis. And again, we have a model for this, which is often later on in the diagnostic process. So I just wanted to really cover, I'm coming gradually to the end of the talk, but really bringing it all together and sort of thinking, how can all these models Aiforia have be used to optimize and enable the workflow and fundamentally change the pace and process of diagnostics. With AI in the loop, we can start to break free from the sort of sequential paradigm and create a more parallel, intelligently streamlined workflow. Imagine when a biopsy comes in and it's scanned, the AI immediately triages the H&E slide. If the AI grading model and the cancer detection confirm, for example, that there's an invasive carcinoma on that H&E, we don't need to wait for the pathologist to identify that it's a tumor. It can automatically order the IHC panel in a reflex-like manner. The system can be set up so as soon as the cancer is flagged, the IHC panel is reflexly ordered or even begun. In practical terms, this might mean that by the time the pathologist is first looking at the case, the ER, PR, and HER2 are already in progress, being processed and queued in the lab. This early ordering could shave a day, maybe 2 days, 3 days off the total turnaround time. Rather than a pathologist seeing the cancer at 5 p.m. and then those stents only starting the next day, the AI would have essentially seen it when they were being scanned and prompted the lab to start the IHC later on in that afternoon. Essentially, the AI triage allows certain decisions to happen in parallel that used to happen in series. Now consider with HER2 reflex in situ hybridization scenario. Normally, if a case ends up as a HER2 2+, a pathologist would then have to order FISH. And that would potentially add a week, depending on how if the lab batches things or whether they do them sequentially. But with the AI Aiforia HER2 model, we could know what the likely score would be much sooner. As a pilot concept, if the AI analysis of the HER2 IHC shows a 2+ pattern with the HER2, the system can automatically trigger the lab's FISH testing protocol immediately. And this means by the time the pathology formally confirms, yes, this is a 2+ and needs FISH, the FISH could already be underway or even completed. We're effectively compressing the reflex loop through the use of AI. This kind of automation could remove a huge chunk of idle time where the case would otherwise just sit around and be waiting for the next step. It's important to stress that any such trigger would be configurable. So labs could set their own confidence level. For example, only auto read if FISH, if AI is very confident and perhaps with a quick pathology review, for example. But if the technology makes it possible to do these things in a smarter way, it could really revolutionize the way the lab is functioning, not only at the pathologist diagnostic level, but also back in the lab with the processing. Another optimization involves cases where the initial H&E appears negative for tumor. Let's say that a biopsy, for example, was taken from a radiologically suspicious lesion, but then the H&E level doesn't show the cancer. Usually, a pathologist would have to manually request deeper sections or additional stains, such as pancytokeratin, for example, to see if there was something, to hunt for the tiny focus of tumor cells. AI can assist there by essentially performing a first pass screen of that H&E for any signs of malignancy. And if the tumor grading cancer detection system didn't detect cancer, it could automatically suggest or trigger deeper levels or confirmatory IHC the same day. Instead of waiting perhaps for another day for the pathologist to ask more cuts after the initial review, the AI could prompt the lab to cut deeper sections preemptively when it saw nothing on the initial slide. This means that by the time the pathologist comes to assign out a case that looks negative, the additional levels are already prepared for the pathologist. It closes the loop faster on cases that might otherwise ping pong between the lab and the pathologist over multiple days with additional work. All of these interventions, early IHC ordering, automatic reflex fish, auto deeper cuts, represent a shift to a proactive workflow powered by AI and powered by Aiforia models. They turn a linear process into a more parallel dynamic process. The workflow becomes AI augmented. Some decisions are made by a smart algorithm in real time, while others are made by the pathologist. But the end result is that each case moves through the lab more efficiently. Of course, we're implementing these carefully via pilot programs to ensure patient safety is paramount. Each trigger is under controlled evaluation. The ultimate vision is that labs can configure these AI-driven rules to suit their needs. For example, a lab might choose to auto-reflex HER2 fish only if the AI and the attending pathology both flag the case as 2+. Another might just do it automatically. The system is flexible. The big picture is that the approach can dramatically shorten turnaround times and reduce those dreaded waiting periods. Instead of a biopsy taking, say, 7 to 10 days to get all the IHC and ISH completed, maybe we can bring it down to 4 to 5 days, maybe even shorter. For patients, that's less waiting time for anxious results. For oncologists, it means treatment plans, especially starting HER2 targeted therapy, can be finalized faster. And for the lab, it means a higher throughput. Cases are not bottlenecking at any one step for a lack of a decision or an order for a test. It's exciting to reimagine the workflow where sort of AI and pathologists are collaborating together, not just making interpretations, but also orchestrating the lab process in an optimal way. And what is the end game really here? To conclude, it's a talk of really trying to develop an AI augmented pathologist, not an AI replacement of pathology, but augmenting them to make them faster, more accurate, more consistent. It means that it's moving the pathology practice, the pace of pathology, quicker. Throughout this presentation, we've emphasized the aim that it's not to replace them, but to empower pathologists. In our envisaged future, every pathologist has an IA-based assistant based on the workflow and the panels of Aiforia models we have, much like a sort of co-pilot, handling the repetitive, time-consuming parts of the job and providing extra insights, while the pathologist can focus on the higher-level diagnostic thinking and the decision-making. When we combine the strengths of AI and the expertise of pathologists, we see a clear path to greater diagnostic accuracy and consistency. The AI can detect subtle patterns and apply quantitative analysis uniformly, reducing the chances of human oversight and variability. Meanwhile, the pathologist's medical judgment ensures that the context and, for example, unusual cases are correctly interpreted. Together, they form a safety net for each other. AI catches what the human might miss, and the human catches what the AI might misjudge. The results improve quality of diagnosis. We foresee a situation where our Aiforia breast suites can make a much more efficient workflow. Many of the pain points we recapped, the waiting times, the manual counting, the repetitive tasks, can be drastically reduced. Turnaround times that once stretched to a week or even more than a week can be compressed down to days. This isn't just speculative. We've seen in our studies that incorporating our Aiforia models lead to a significant time saving, 50% faster grading, 40 to 50% faster with scoring in the IHC panels. In practice, that means that labs can handle more cases in the same amount of time, or give pathologists more breathing room and reducing burnout. An efficient workflow also directly benefits patients and clinicians by again getting results out faster. Imagine biopsy results reliably coming out around 48 to 72 hours instead of a week or more. That kind of improvement we're aiming at by automating all the steps and enabling parallel processing. Crucially, this pathologist-centered approach, the pathologist remains at the helm of the diagnostic process. All the AI tools and the AI models we have discussed and shown that Aiforia offer are clinical support systems. They provide recommendations, measurements, alerts, but the final interpretation and the ability to edit the results remains with the pathologist. In our design philosophy and regulatory approach, we ensure that pathologist is in control at every aspect. They can accept, modify, reject the AI findings, much as you might do with a colleague when you ask them to look at a case with you. The AI is there to augment your capabilities, much like how modern pilots rely on autopilot for routine flying, but take charge during takeoff, landing, and if anomalies arise. This approach is intended to maintain trust, both the pathologist's trust in the tool and the clinician's trust in the pathology report. By keeping pathologists as the ultimate decision maker, we also safeguard the speciality of pathology. We don't want pathology to become a commodity, digitally driven, just number-based topic. We all understand who see slides that it is a speciality and it needs to grow in the framework of utilizing AI. Feedback from early users of our systems and pilot studies have been really encouragingly positive. Pathologists who have tried the AI tools often report that they feel more confident in their diagnosis and appreciate the second set of eyes. For example, in our usability evaluations, we have found that they feel more and more assured about the results when they're signing out. They also reported that it streamlined their workflow. Less time on the boring grunt work meant that they had more time to think about a case as a whole, or even handle additional cases without feeling overwhelmed or cognitively overloaded. Several have said that it's like having a diligent junior colleague or even an experienced colleague who never gets tired. Of course, they also give valuable feedback on how to improve the tools, and we take this constantly. Ultimately, our grand vision is that embracing AI will lead to better outcomes. We're not just giving diagnosis. Every diagnosis has a patient behind them, and we want them to get the best treatment in the most timely manner, and we want to improve better outcomes. We essentially want to provide results at higher speed and better consistency, which will ultimately help improve patient care. Faster, more accurate diagnosis means treatment can be started sooner and tailored better. Consistent interpretations means that patients can trust the results will be the same no matter where they are reviewed. And easing the workload on pathologists means the specialists can devote more attention to complex cases, research, and other consultations, further elevating the quality of diagnostics and the interest in the work they're doing. We believe that an AI augmented pathologist is not just this thing in front of us that's maybe in 10 years, but it's actually something that we're building now with you pathologists, us in Aiforia, in collaboration, with a grounded clinical approach. By working together, Aiforia, clinicians and researchers, we're on a path to a new era of pathology that retains all the rigor and insight of traditional clinical practice, while shedding many of the inefficiencies and variabilities. It's an aspirational vision, yes, but one that is very much within the reach, and we're excited to be making this reality with you. And that's all I've really got to talk about. I hope it's inspired you to look more into how AI can make your practice more efficient and more interesting and more exciting. And now we're open to questions.
And yeah, of course. Now it's time for the Q&A session. Let's start with some questions. And if I look at the Q&A button of our chat, there are already some questions in. So let's start with those. Can the AI help with prioritizing which cases I should look at first? Richard, can you please give an answer to this question?
Yeah, it certainly can. So when the slide is scanned, for example, it what basically generates you get in the clinical viewer, you will get a clinical list, and it will say what the type of sample is, for example, breast, and it will say if invasive carcinoma has been identified or not. So, for example, presumably, if you want to focus on the cancer positive cases, you can use the initial clinical list to click and choose that you will have at the top of your list invasive carcinoma, so you can focus on those, get those out, get maybe ordering the stains as quickly as possible. So yes, you can use it to prioritize, essentially triaging your cases. Absolutely. And that's one of the advantages. It's very nice in high volume laboratories where you may have a stack of cases. You never know whether or not the first case will have the cancer, the last case will have the cancer, or in the middle. What this Aiforia process, which identifies the tumor, of the cases that have tumors before you see them, you can use it to identify those. So you can essentially find all those cases within, let's say, the 100 cases, let's say the 25 cases of cancer, you can find them, put them top of the list and start focusing on them. Yeah, it's one of the advantages of using the system.
All right, thank you, Richard. I have one more question for you. How much control does the pathologist have when using the AI models?
Yeah, essentially with the Aiforia system, the clinical viewer, that is one of the things that we've been really emphasizing, and in the development with clinicians, this sort of hand-in-hand development, we've made sure that the pathologist always has full control. Firstly, the pathologist can always overwrite the results in the results section that will go over to your LVMS system. So the pathologist can always change it if they want to. But they also, and this is very unique for us, can actually interact with the image, take areas out that they think, for example, if you had an area of artifact that you wanted to take out, that you weren't sure, should I include this or not, you can actually edit that result. You can draw around it as you would in, with an art function, and then just remove that area. You can also, if there was an area you were particularly interested in, for example, in breast cancer, breast pathology, there's becoming an increased interest in heterogeneity of cancer, where certain areas express certain of the biomarkers. You could, in fact, use that function to circle around a particular area and see what percentage is this, what grade would this be with the grader, or with the ER/PR HER2, you could circle around and see what percentage is this area HER2 positive or ER positive, for example. So one of the really central tenets of the Aiforia system is that the pathologist can interact, can work with it, can change it if they need to. And I think that was a lesson that we took over from what has been seen in radiology, where they didn't just want something that gave them a result, gave them a readout, but they wanted to actually be able to interact. That interaction can take some time. It's a bit like learning anything. So initially you can be overwhelmed when you see it, but if you keep using the system and learn the functions, eventually it becomes almost second nature to use this ability to interact, change the result, override the result. Yeah.
Great, thank you. We have one more question. Actually, we have three more questions. And one of them is there an AI model in development for hair-to-ish or fish interpretation?
Excellent question. And I was tempted to put a slide in. We do have one customer who got us to custom build a HER2-ish model, which is actually functioning very, very well. It's not currently on our roadmap, but if there was demand enough for it, we will. It is in our roadmap, but not in the near future. However, if there was demand, we would certainly do it. The customer that is using the HER2-ish model is very, very satisfied with it and is using it. And as we all know, it's a very laborious thing to count HER2-ish, and it takes some time, and it's not the most exciting thing to do. So automating it is a good thing, and we have seen with that customer that they like the use of that. Also, one of the advantages we can have is that if you have a HER2 positive identified on IHC, it is potentially possible to automatically order the ISH before the pathologist sees the slide. So, and that will, as we know, ISH can take a lot of labs batch this analysis. So if you reach it when just the batch is gone, it may take another week or 2 weeks to get the ISH result. So then you may take a bit of, it will further delay the diagnosis. So by having this automated ordering of ISH from the HER2 IHC, it could really make it effective. And if we had, as you say, the HER2 ISH model, that would make it even quicker. So yes, it's on our roadmap. We have made a research use only for a customer, but we don't have it currently available to the wider audience. But if that was demand and you as customers were telling it this would be even greater and really be very useful for us, then it is certainly something we would look at and probably take up earlier in development.
Thank you so much, Richard. This is the end, I'm afraid. Thank you all for your questions. And for those who would like to explore further, please visit our website and feel free to reach out. We'd be happy to continue the conversation. A big thank you once again, Richard, and for the excellent presentation, and to all of you in the audience for joining us today. I wish you all a great rest of your week. Bye-bye.
Thank you. Goodbye.